Good morning, everyone. So yesterday we heard from Veronica how to make thin lamellas from frozen samples. And she mentioned many times that those samples have been high pressure frozen. And so today we are going to talk about that technique, what entails to high pressure freeze cells. And so we're going to talk about directly what high pressure freezing is. Then we are going to discuss a couple of options in the market for high pressure freezers, and then specific carriers and applications for the different type of tools. And so first of all, we need to freeze our cells before we can do anything else, right, for cryo-electron tomography. And so as we all know, the main challenge that we have is to freeze cells without destroying the ultrastructure. We want to keep the ultrastructure protein complexes membrane as close as possible to the native state. And that's a challenge because as we are freezing our samples and water is cooling down, it will start to crystallize. And as it crystallizes, it will expand, it will increase volume. And those growing crystals are going to displace our molecules, the cytosol. If they are big enough, they can break membranes, organelles. And so we cannot just freeze slowly a sample without compromising the cellular integrity. One option is to cool them very, very fast. So if we can manage to get a cooling rate of around 10,000 degrees, it will cool so fast that the growth of those ice crystals will slow down, or they won't have much chance to grow, and we can then vitrify the frozen water. It can be obtain an amorphous vitreous ice that it won't disrupt the cellular structure. And those freezing cooling rates can be achieved by using liquid nitrogen or liquid ethane, and that's pretty much what we do for plant freezing, right? So we just have your cells on a support grid, and you plunge it into a liquid ethane, for example, and if the cooling rate is quick enough, you will get good vitrification of your samples. Now, the problem is that the heat conductance of water is not that great. It's really very poor. And so although we may be able to achieve those cooling rates on the surface, as we are moving into the volume, it could be water volume of your sample, then those cooling rates will start to slow down. And what they do, the ice crystals will start to grow again. So in practical terms, it's possible to do rapid freezing without ice crystallization on samples around 10 microns or smaller, but for bigger samples, it's going to be tricky, and you risk the fact that you may have ice crystal damage within your sample. So here comes high pressure freezing. If you freeze them while applying high pressure, then we can vitrify water, we can obtain that amorphous ice, and the ranges of pressures that we are talking about are around 2,000 bars, so 2,000 times the atmospheric pressure. 2,100 bars is what most high pressure freezers uses, so we can supercool the water and freeze it in an amorphous state. So this is basically what we are talking about. So this diagram is showing us how, from temperature going down, and here the pressure going up. So if we try to freeze at atmospheric pressure, we are going to get hexagonal ice 1. This is the bad one for us, crystalline ice. But if we increase the pressure around 2,100 bars, then we are going to be obtaining ice 2 and ice 3, and here is just a little diagram. So these are denser type of ice, amorphous type of ice, so they are not going to expand, and they are not going to disrupt as much our cellular structures. So this is done by lowering the freezing point. You can see that we are supercooling the water by doing that, so we are increasing the density of the water as we are doing that. We are reducing the rate of ice crystal nucleation, and we are slowing down by applying pressure as those ice crystals are trying to grow. The pressure will restrain that growth and keep, if they are ice crystals, they are going to be very small. So that's the principle of high pressure freezing. We use pressure to avoid ice crystal formation. And so this is a graph from the Leica ice high pressure freezer that some of you have been already trained on. And so what it does, and you can see here, there are two curves. One corresponds to temperature in blue and the other one to pressure in red. So by the time that we are ready to cool down the sample, the pressure has been raised to, again, around 2100 bars. And then the temperature goes down very, very quickly. And as you can see here, the scale, this is in milliseconds. So what a high pressure freezer has to achieve is this increase in pressure and cooling rate very fast in the range of 20 milliseconds. That's what is required. And if that's done and that is properly done, then water can be vitrified and hopefully our samples as well. Now there is another challenge. And this graph is showing us the cooling rates in a high pressure freezer. So this is done under pressure. We are already in a high pressure freezer. And so what this graph is showing us is from the surface. So this is the surface of our samples or our volume, going all the way down to 100 microns into our chamber. And this is time. So you can see that from the surface, very close to the surface, we can get very high, quick cooling rates, the 10,000, this is kelvins per second. So in this range, very close to the surface, we are cooling very, very fast. But as we are moving into the sample and farther away from the surface, those cooling rates now are slowing down again. And that's the challenge, how much farther you can go. Well, you can go to 200 microns, and then they are slowing down enough to allow ice crystal formation. So there is a limit on how big the samples can be in a high pressure freezer anyway, and it's around 200 microns. So what happens if ice crystals form? Again, hexagonal ice will grow. It can disrupt membranes. It can pull away material. So this is not cryo electron microscopy. We will discuss that. High pressure freezing was actually developed decades ago for regular TEM. So these are samples that have been contrasted with heavy metals, but it's a good example to show what is ice damage in a high pressure frozen sample. So this is chromatin within the nucleus. When we have ice growing, you can see that, well, let's just start with a good sample first. So if the ice has been vitrified, then we will have a very homogeneous structure. But if we have ice growing, you can see these little empty places, right? So this is where all the ice crystals were growing and pushing away the biological material. It can be very subtle, or it can be really bad. It depends how big those crystals were able to grow. So another example of good and bad, maybe you can guess which one is good and which one is bad. So in the top one, we have, this is, what is this? Golgi. And so you can see here the membranes, you can see the bilayers, right? We see ribosomes, a nice homogeneous cytosol. But when we get into this sample, you can see that the membranes are disrupted. You can see again these kind of empty areas where ice crystals were growing and pushing the material all around. So, and these are from the same sample, and the pictures were taken by Ashish. So you have to be careful that within that chamber that we are going to be freezing is a very heterogeneous environment. As the bigger the chamber is, the more heterogeneity you will find. And exactly because those curves are showing us that the freezing quality is not the same all inside that volume, So it is important to keep in mind that you may have good cells, bad cells, and you need to optimize your protocol before you go into cryo-feeding, because again, you don't want to end up with a sample like this or just a bad cell. Other things to consider, we said that 200 microns is the limit, but actually depending on the sample, you can go a little bigger. So there are many cells or many tissues that contain natural cryoprotectants. So if your tissue is very rich in glycerol, sucrose, or some small sugars, oligosaccharides, they're gonna protect your sample from ice crystal formation. And so you can actually get to 500 microns or even higher. It depends on the sample. One issue with high pressure freezing is that you are applying 2,000 atmospheres of pressure. If there is any risk for having gas bubbles or air bubbles within your sample, you can imagine what is going to happen, right? So it's going to collapse that bubble. It's going to restrict the pressure, right? Because it's being dissipated by the bubble. So you have to be extra careful that there are no gases, no air bubbles inside that chamber while you are freezing. And consider using a cryoprotectant, not to get inside your sample, but you also have, most likely, because you don't want to leave any empty space or any bubble inside, you are using buffer, you are using a cryoprotectant, and so it's important to keep that medium without ice, crystalline ice. So you also want vitreous ice outside. So you may want to consider using, adding to your buffer a cryoprotectant like dextran or glycerol. Okay, so I mentioned high pressure freezing has been used, and it's been used for a different, for a wide variety of imaging techniques. And so the images that I showed you before, so once the samples are frozen, they can be dedicated to conventional TEM. And that's why high pressure freezing was developed in the first place. So then the frozen water is replaced with solvents and fixatives at very low temperature. They can be embedded in resin at these very low temperatures, -60, -50 degrees. And then once those blocks are fully those samples are fully embedded in plastic resin, then they can be sectioned for a regular conventional TM, being immunolabeling or cellular structure. You can use it for correlated light and electron microscopy applications. They can also go to electron tomography at room temperature, or they can be used for the different modalities of block phase SEM as well for large volume reconstruction. Maybe your frozen samples can also be cryosection, and we will talk about that, in which cases we may want to do that directly for cryo-ET or for some other applications. And then, of course, what we have been discussing during this workshop is cryo-feeding and getting very thin lamellas out of those well-vitrified cells for cryo-ET. So this is pretty much the pipeline that we're going to be discussing, right? From freezing, how we get into the cryo-feed, that it was what Veronica was telling us yesterday, and then for cryo-ET. Okay, so that's why we use high pressure freezing, when we have big samples, big cells, and we want to minimize ice crystal formation. So let's talk about the options on how we can accommodate our samples. So Veronica also was mentioning these freezing planchets. They can be made of copper, aluminum, and they come in different shapes. And some of you have been freezing some of these planchets in the past 2 days. So they can be 3 millimeter in diameter, 6 millimeter in diameter, and they have different shapes that allow you to create different chambers, different volume chambers for freezing. So there is a type A that in each side, it has kind of a well, so it's 0.1 millimeters on one side, 0.2 millimeters on the other side, that's the depth. The type B is 0.3 millimeters deep in one side, and then type C, the same depth in both sides, 0.15 millimeters. And so combining the type A, B, and C together, you need to put one on top of each other, so they are always working as a little sandwich. You can create chambers of different sizes, and you will fit your samples inside. So you can create chambers of 0.1 millimeters or much bigger. You can also use spacers between the freezing planchets if you want to create a specific size of your chamber. And so these are all options for all high pressure freezers. You can use these freezing planchets. There is also the possibility to use sapphire discs. So you may want to grow your cells directly on a sapphire disc. So for example, here we have some mammalian cells growing on a sapphire disc, so it can be carbon coated, so you can print a pattern that it will help you to do correlative imaging. So you can image those sapphire discs with your cells before freezing. And then again, using either spacers or the different type of freezing planchets, you can create these sandwiches and place the sapphire disc inside. And then, as you probably have done during the practice, you will detach the two freezing planchets and recover your sapphire disc with your cells. And this is very useful for correlative work. So either you can image before freezing, so you can see which cells are expressing your fluorescent proteins, which cells are expressing your fluorescent proteins, or they are in the process that you all care about. You freeze them, and then you can locate the same position. So if you're going to be doing cryofeeding, then you know where to go. And so at different points during the process, you can locate the area. So working with sapphire discs facilitate correlative CLEM pipelines. Other type of carriers. So these are dome freezing hats. So when you put them together and then freeze something inside and detach it, it will create a dome. So your cells are here in this ice dome. And that can be then taken to a cryo ultramicrotome for sectioning. There are some carriers that are just tubes made of copper of gold. So if your sample can fit into this very narrow tube, is cell culture, for example, or yeast, you can place it inside and freeze the whole tube. And then you can section the metal tube and expose your samples. So why we may want to get into this? So cryo-ultramicrotomy of vitreous eyes, or CEMOVIS, is a technique to section vitrified samples. It was very popular when there was no cryofib. That pretty much was the only way to get a thinner lamella. Some people still may use it to get, let's say that you freeze something very big and to go to the cryofib, you want to even get something slightly thinner so you can get a thicker slab in the cryo-ultramicrotome. I think that is good news that the cryofib is taking over, because this was a very difficult technique. So what we are seeing here in this movie is sectioning that frozen block. So you can imagine all the things that can go wrong. From ice contamination, you don't want any crystal to form on those sections. So a ribbon of sections through that vitrified sample is collected. There are micromanipulators, so Leica sells a micromanipulation system for their cryo-ultramicrotomes that allow you to do this. And then you can collect all these sections on a grid. And again, it can go directly into imaging or any further processing that you may want to do. Now, it's not only very difficult to do, but it also brings some, let's just stop this here. It also brings some artifacts with it because this is ice. And as you are sectioning through, it can induce some artifacts like compression. So you can see the direction of the cutting. So the knife was oriented in this direction. And as it's moving through the knife, the block is being compressed. So it's not only a very difficult technique, it also brings some artifacts. So it's not the most favorite technique to use if you're trying to get thin lamellas for cryo-ET for all these reasons. Okay, so let's talk about different options for high pressure freezers. So the most popular and the only one that was available for many decades was the HPM 010. It was first commercialized by Voltec. Abra Valoinen now is selling the current model. And have any of you used these machines before? So they were great. They freeze very well, but they're not very practical. And so what you can see here is the sample holder, and this is the tip of that holder. So you prepare the sandwich of the two freezing planchets outside the freezer. We have another station to do that. And then you put it here, you screw this tip, and then it goes into the machine. And here we have a movie that I borrowed from Scott Robinson. And so you place the holder there, then you put that bar because otherwise it will shoot through the roof. And then freeze it, and you have to take it manually out whenever you think it's ready and plunge it into nitrogen so it keeps the same temperature. So it freezes very well, but reproducibility is not that high, right? Because as you take a little bit longer, it's cooling, and it doesn't read very well. Depending on the models, they cannot read pressure and temperature while you are freezing. So these were great, but they have some limitations. The Leica ice, that is the one that you have been training on, it has a very different concept. So the loading of the freezing planchet is done in the same machine. So it has a dissecting microscope included in the design. And so you have the loading station. And so you can use the 6 millimeters, the 3 millimeter carriers. But what it's adding here that it was not necessary for the previous design is these carriages. So there is a bottom part, it's like a cylinder. And if you haven't seen them, you will see them this afternoon. And the top part that goes in the lead. And then there is a middle plate where the carriers will fit. And then the only thing that you do is just close that lid, and the machine will do the freezing for you. So there is nothing like, manipulating. And I just found this movie online. So we don't see what is going on inside. You will just close the lid and magic, the carriage with your freezing planchet will drop into a container. But this is what the machine is doing inside. So you load it here, right? So you close the lid, and when that happens, then there is this arm that is gonna go in and take the carriage with your samples inside. It happens very quickly. You hear all the noises, but you don't see what is going on. And it will introduce in this chamber where the pressure and the liquid nitrogen jets at high pressure are injected to your hats. And then it will drop the carriage with your samples in this container that then you need to remove. So it's very reproducible. And for each run, it will give you a reading of the temperature and the pressure, so you know exactly what the conditions were. And it comes with a number of accessories and options that weren't available for the previous freezers available. So, for example, there are chambers that work with sapphire disc, where you can grow your cells, you can do your live imaging, and then these can be quickly transferred into the sample loading station very quickly. I haven't done it. They claim that it can be done in 5 seconds. I don't know if that's true. But that's the idea, where you identify a cell and you know in your Sapphire disk which cell is doing something interesting to you. You can freeze it very, very quickly. There are some accessories where you can do light stimulation and electrical stimulation to your sample before freezing. So that means that you could be activating certain processes. Electrical stimulation has been used for neurons as well. And in a very short period of time that you can control, you can freeze them and then follow a given process, like fusion of vesicles to a membrane or the transmission of a signal. And then after the samples are frozen, there are a number of applications that they are used for. So for freeze substitution, again, is the replacement of water by solvents and resin at low temperature. It can be done for morphology, for immunoglobulin labeling, or for CLEM studies. And Semovis is the sectioning that we already discussed, cryofib, of course, and freeze fracture. And so I hope that everyone will have the chance to give it a try and see how it works. And that's all I have. Thank you.
Thank you, Marisa. That was fantastic. Questions? You got that one? Okay.
That was a great talk. I have a question. So you mentioned the sample thickness is 200 nanometers for high pressure freezing?
Microns.
Microns,
Microns.
Okay.
That would be difficult.
I agree. That was a really good talk. I'm wondering if you are growing cells on those Sapphire discs. Do you want to grow them in the planchette? Like, do you add your media in like the well and then turn the disc over and grow the cells on the planchette? And then does it really matter about directionality when you?
So when the cells are grown on the Sapphire disk, you have them in a cultured dish. So the planchets are copper and brass, and I wouldn't grow cells there. So you can grow them in any medium that you want on a plate, and then you just transfer them into the planchet just before freezing. And the design, you can design a chamber, a small chamber on top, and then a flat, like the type Bs have a flat surface. So one common design is to have a flat freezing planchet, the Sapphire disk, and then a type A with the small chamber facing the planchet. So you are going to get like 100 microns ice on top of the Sapphire disk. You can do it with the spacers. So maybe you want to have 2 type Bs, like flat and flat, and then a spacer giving you exactly the thickness that you want for the Sapphire disk. And it's kind of the principle of the waffle method that we are going to hear in a couple of hours. So you have a lot of different modalities depending on what you want to do and how much ice you can tolerate.
Okay, thank you.
Sorry, that wasn't my question, actually. So my question is that if it's 200 micron, right, and then your sandwich, some of them can get to like 450 micron. So how are you going to make sure that they can freeze like well?
So again, it's 200 on each side. We have frozen things that are quite big that almost don't fit into the chambers. But it's always from the surface where you get your better freezing. So it depends how homogeneous you need something to be. We do mostly high pressure freezing for freeze substitution and TEM. And when we do that, we can check the quality across the sample. If you're doing cryofib, I think that it's very critical that you are sure that it's optimized as much as possible because you don't have the chance to check before you go into making the lamella. So the thinner your chamber, the better, always. And because even if your sample can freeze well, there will be much more heterogeneity. So, no. It's always good to work with the smaller volumes than with big ones.
Hi, I just wanted to ask quickly, how do you get the sample then after frozen to a grid? Is the grid in the planchette, or where? How do you get this to an actual TEM grid after freezing?
Okay, so if it's a frozen planchet, it depends on which of the modalities, right? But if it's a freezing planchet, and we are going for cryo-feeding, so Veronica was showing us, right? You can do the cryo-feeding inside the planchet, or you can do the cryo-feeding on the sapphire disk. So they can stay there. And again, we will see in a couple of hours another technique where you use a freezing planchet to make a sandwich, and you freeze samples on a grid that then can be taken to the cryo-feed. So people are very creative about making little sandwiches and devices to have very thin layers of ice that then you can take to the cryo-feed.
There are two online questions. The first one is, when, after you've frozen with HPF, might you have indications whether you've created a good sample, that it has good or bad ice? So suppose you're going on to like a FibSim. Would that be a step where you might see that?
Difficult, but for example, sometimes you can see something that happens. So you remove the freezing planchet, and let's say that you were using a flat B on top of an A hat, and you see that there are holes of, the sample is not homogeneously frozen, I wouldn't use that. Obviously something happened, or there was air, or some of the samples flew out of the hat. But otherwise, it's very tricky to see. So one possibility is also to try to optimize freezing and do freeze substitution. First, you check how your samples look like. You become very familiar with this, a good freezing, and you optimize your protocol there. And then once you know it's working well, you go to cryo-feeding.
Well, that was a great explanation for that. And our next question was also, could you share some examples of organisms that you'd recommend or that you think are really great targets that people might be looking at HPF as a technique to be applying?
Yeah. So again, anything bigger than 10 microns should probably give it a chance. In my lab, we work mostly with plants, but we collaborate with many other researchers. We have done C. elegans, yeast, Drosophila embryos, and again, in plants, pollen grains. We are doing Chlamydomonas, roots, leaves, anything that you can fit into those planchets is worth of giving a try. If they are tissues and there is air, you may want to get rid of that air before, but otherwise, pretty much anything can be frozen in this way.
I had two questions. You had mentioned like trehalose and other cryoprotectants. I've also heard of like methylpentane and hexadecine. I was wondering if you have any thoughts about when those are useful, if it's just purely empirical. And my second question was, one thing we've struggled with is getting the hats separated under in liquid nitrogen. I was curious if you had struggled with that and then how you had overcome it.
Yeah, so hexadecine is used as a cryoprotectant and also help you to detach the two hats. That's the preferred method for doing that. Other cryoprotectants, it also depends what it comes next and what is compatible with, cryo-feed milling or cryo-ET. But solutions that contain sugars are good, or BSA. It depends what you are doing. Some people use yeast-based, just a lot of yeast around your pressure cell, and that will help to keep an environment without ice. Trehalose and glycerol, those are natural cryoprotectants. And so if your cells contain a lot of that, you have better chances of getting good freezing. If there is a lot of water in those tissues or on those cells, as we face with plants, then there is not much help from the internal tissues, and it's just water, heat conductance, and we have more ice. So there's a lot of variation, and that's why it's important to optimize for your own cell what works best before getting into the cryo-feed milling pipeline, to be sure that you have optimized as much as you can the freezing conditions, cryoprotectants, and so on.
Just as a follow-up, there was a question in one of the practicals for hexadeciane. When you plunge freeze it, or when you high pressure freeze it, and then you put it in the TEM, does the hexadeciane like sublimate, or what happens?
We haven't. There are some other solvents that are used, and actually some of them can sublimate before you go, so you have a clean slab. I don't know with hexadeciane what we haven't used it. Question.
Other questions? Any more online, Matt? Or oh.
I think we could probably do one more of the online questions. The question is, what parameters are there to optimize for HPF besides sample thickness and the cryoprotectant?
So with a machine like the ICE, there is not much. With the Voltec and Abra machines, there were other things you could optimize. That those machines use ethanol for creating the pressure, and you could check the pressure, you could change the pressure, and that would change a lot the quality of the freezing. The ice is more like, just put it there and there's not a lot of things that you can change, but certainly the type of hut that you use, if you're going to use spacers, if you think that it's too thick. So all the practical aspects of the environment, the cryoprotectant, but that's pretty much it. The conditions of the machines are not something that you can change easily.
Okay. All right. Let's thank Marisa again. That was really wonderful. And